So next up is Sue Liebman, who's going to tell us about prions.
You can fold the clothing. This is on? Yeah. No? Okay. So I'm going to tell you today about a journey from basic yeast genetics to yeast prions to protein misfolding disease. And before there was direct DNA sequencing, Fred Sherman devised a way to determine the sequence, the DNA sequence of the iso-1 cytochrome C gene. And he did this by sequencing the protein of intragenic revertance. And he used this system, as we already heard, for studying translational initiation, but he also did it for many other things, including showing that the genetic code in bacteria was the same as that in the eukaryote yeast. And we heard about the cloning of transfer RNA, but he did this, he used his deduced sequence to help clone the first yeast protein coding gene, and for the study of informational suppression, which is what I'm going to continue talking about. So by isolating intragenic revertance of a single mutant, he noted that each one of these inserted amino acids at position number 2 had the E. coli predicted codon that was one base pair removed from the UAA stop codon ochre, identifying this as an ochre mutation, and similarly other mutations as ochre and amber in iso-1 cytochrome C. And this identification was key to our isolating a variety of nonsense suppressors that misread these nonsense codons, anti-suppressors that reduced the efficiency of misreading of nonsense codons, and allosuppressors that enhanced the efficiency of misreading these nonsense codons. I'm just going to expand on Anita's comment about the tRNAs. The reason Don knew that they were tyrosine tRNAs was because we showed that they inserted tyrosine at position 2 at the iso-1 cytochrome C nonsense codons. These were all Mendelian suppressors, but in 1965, Brian Cox described a non-Mendelian, or should I say, unclear genetic factor, and this was an allosuppressor, and he showed that it was cured by guanidine hydrochloride. And some years later, Fred and I showed that it could also be a suppressor on its own. When I started my own lab, I started working on actually allosuppressors and anti-suppressors of the SUP35 and SUP45, and didn't generate a lot of interest after a while from NIH, so I switched to something sexier and started working on ribosomal RNA, and that attracted Yuri Chernoff to come and join me as a postdoc in my lab. And he brought with him a project that he had started on looking for anti-suppressors of Psi. And he was in Bun Ono's lab in Japan when he did the beginning of this work. And one of the clones that he pulled out was the HSP104 gene that in high copy called loss of Psi. And since we were in Chicago down the street from Sue Lindquist, who had a bunch of HSP104 mutants, we talked her into working with us and getting some of her mutations. And together we showed that either overexpression of HSP104 or deletion of HSP104 caused loss of this esoteric Psi factor. And we were writing this project up. We figured at least maybe 5 people in the world would be interested to read this paper. But just then I got a telephone call from Reed Wickner. And he told me that prions were real and that not only was Uri3 a prion, but Psi was a prion. And he was a prion of SUP35. So now our chaperone effect on Psi became strong support for the protein-only hypothesis. We now know that the SUP35 is composed of a prion domain, and the functional translational termination factor region, and key support for the protein-only hypothesis was that transient overexpression of the prion domain causes induction of psi, and that the prion domain is required for the maintenance of psi. So SUP35 prevents, the release factor prevents the ribosome from reading through stop codons, and if there's an add1 stop codon mutation, cells are read. In psi minus, however, the SUP35 release factor gets attracted away from its job and joins the psi seeds, and therefore there's a read-through and the yeast become white. Psi aggregates grow from their ends, and therefore they require something to shear them so that they generate more ends and can keep growing, and that the small aggregates can segregate to the daughters. And in the absence of HSP104, we now know that this doesn't happen, and therefore HSP104 is lost. Overexpression of SUP35, transient overexpression of SUP35, causes the permanent appearance of psi, and we found that we got different, we call them variants, we'll call them types now, types of psi. And for example, the weak psi turned out to have larger aggregates, therefore it had fewer ends and more soluble functional SUP35, so the cells were less white than the strong psi. The finding was analogous to the finding of mammalian PRP strains that cause different pathologies of illness, and these had been considered to be a problem for the protein-only hypothesis because they were suggestive of cryptic viruses. So the yeast result provided support for the protein-only hypothesis. And we now know that other protein misfolding disease-associated proteins also can propagate as self-seeding conformations with distinct disease properties. To show that these different psi types were not due to a Mendelian modifier, we began, for example, with weak psi, cured it of psi on guanidine hydrochloride, and then transiently overexpressed SUP35 to reinduce psi, and we found, as expected, that we got both weak and strong psi back. However, surprisingly, some of the psi minus that we isolated, no matter how much we overexpressed SUP35, we could not reinduce psi. And eventually we figured out that was because there was another prion together with psi that was sometimes lost, and then you couldn't induce psi, and was sometimes not lost. And so we called this PIN for psi induction, and we found that it was required for the induction of psi, and it was the prion form of the RNQ protein. And we now know that in addition to these yeast prions, there are other yeast prions as well. The proof of the protein-only hypothesis was done in yeast before it was done for the mammalian prion PRP, and in this rendition, and these proofs used the different psi variants and the PIN factor. So in this rendition, a portion of the SUP35 prion domain fused to GFP is seeded to form a fiber by seeds isolated from weak psi strain, and when these seeds are transformed into psi minus, PIN minus yeast spheroplasts, they gave rise to a large percentage of weak psi. The same experiment done using strong psi seeds gave rise to strong psi. So now a transition. Both yeast prion and neurodegenerative disease associated proteins are often misfolded into amyloid or other irreversible aggregates, and thus yeast prion insights have had a significant impact on the study of neurodegenerative disease. And here's a list of some of these diseases and the associated protein misfolding, protein that's misfolded. I want you to note that TDP43 is associated with quite a number of these diseases with distinct pathologies. Both TDP43 and FUS are stress granule proteins. They are not found in yeast. They aggregate in patients' neurons, in nuclei, particularly cytoplasms, and they're associated with toxicity. They kill the neurons. Several groups modeled ALS by overexpressing TDP43 and FUS, and they found that they formed aggregates and were toxic. And Since they were toxic, they were able to isolate extragenic modifiers in yeast that altered the toxicity. And my favorite modifier was isolated in Aaron Gittler's group, and it is the PBP1 protein, which is a stress granule protein and a homologue of the human ataxin 2 protein. And reducing the level of PBP1 in yeast reduces TDP-43 pathology, reducing the level of ataxin 2 in mice that are modeling ALS by having TDP-43 toxicity also reduces toxicity there as well. And furthermore, Aaron showed that ataxin 2, which has a poly-Q repeat, the length of the poly-Q repeat in ataxin 2 correlates with a risk of ALS in humans, so that a modifier found in a yeast model system predicted a human disease risk factor. And therefore, the yeast model is relevant for human disease. This is not the only case where a human disease risk factor has been identified using a yeast system. So here are some other such examples. Just as PIN affected the aggregation and toxicity of yeast prions, we found that the PIN prion affected the toxicity of overexpressed TDP-43. It's more toxic in PIN plus than PIN minus, and we have more small aggregates in PIN plus than in PIN minus. The PBP1 rescues toxicity, as Aaron showed, and we find that PBP1 deletion reduces the level of aggregates, both in PIN plus and PIN minus, particularly the level of small aggregates. Although the HSP40 chaperone cis1 was not found in the unbiased screens for modifiers of TDP-43 toxicity, we found that just as it affects the toxicity of yeast prions, and just as it affects the aggregation of yeast prions, that it can rescue, when overexpressed, the toxicity of both TDP-43 and FUS. And we found that the level of small dot aggregation of TDP-43 is affected by reducing the level of HSP40. So again, the appearance of these small dots seems to be associated with increased toxicity. And our collaborators showed that overexpression of the mammalian homologue of the HSP40 caused a reduced toxicity of TDP-43 in cortical neurons or of FUS in HECT cells. We don't know why TDP-43 or FUS are toxic in yeast, but we do know that they inhibit the ubiquitin proteasome protein system degradation system, and that overexpressing HSP40 restores that degradation. So now I'm going to switch to another protein, the CREST protein, that has recently been shown that mutations in CREST can cause ALS. And CREST has a prion-like domain and forms aggregates in flies. And yeast does not have a CREST homologue. But just curious fact, the most similar protein in yeast to CREST is the PIN protein RNQ1. So we find that CREST forms nuclear dots in yeast, an occasional cytoplasmic. That PIN, it's more toxic in PIN plus than in PIN minus. It is, the toxicity goes away in an HSP104 deletion, and so does the aggregation. It's now just diffuse in the nucleus in the presence of the HSP104 deletion. Just as deletion of PBP1 rescued TDP-43 toxicity, it is rescuing to some extent, as much as it did for TDP-43, the CREST toxicity. And the level of PBP1 determines the level of aggregation. So in a deletion, there's much less aggregation, and in overexpression, there's much more aggregation, a lot more cytoplasmic, and these all co-localize. The PBP1 co-localizes with the CREST. And our collaborator showed that a reduction in the Drosophila ataxin 2 gene also rescued CREST toxicity of fly photoreceptor neurons. So what I've told you today about the yeast contributions to the prion story is that chaperones are required for prion maintenance, that one prion promotes the heterologous appearance of another prion, that there are different self-seeding conformations, and that purified aggregated protein is infectious. And similar results are now emerging for protein misfolding disease aggregates. And I told you that toxicity is reduced by overexpression of chaperones, for example, the HSP40 and HSP104 chaperone. That toxicity and aggregation are enhanced by a heterologous prion. I gave the example of PIN, and also they are toxicity and aggregation was enhanced by the stress granule protein PBP1. Now there is data emerging for other protein misfolding disease proteins that one protein, so for example, A beta forms A beta amyloid more frequently in the presence of diabetic islet amyloid. Okay. So now I want to end up by talking about what I see as the future of using yeast to look at protein misfolding disease. I think it's a bright future. It's exciting that since these proteins are toxic in yeast, the yeast system can be used to try to identify the actual toxic species. So there's a lot of different species. We have oligomers, reversible liquid droplets, and irreversible solid aggregates that are present. In this cartoon that I've borrowed from a 2017 review, the liquid droplets, which are reversible, the proteins go in and out of that if you do FRAP, is shown as sort of a precursor to the pathological irreversible aggregate that if you do FRAP, it's solid as a rock. And he lists this as being the pathological aggregates. But actually, we don't know that these are the pathological aggregates. It could just as well be that these liquid droplets are the pathological aggregates or the oligomers. So yeah, these structures are all found both in vitro, in humans, and in yeast. We can ask what triggers the formation of this toxic species, and one approach is to ask, how does PIN work? It's been hypothesized that PIN is associated, PIN works by cross-seeding heterologous amyloid to aggregate. That's one possibility, and there's some evidence for that. But it's also possible that PIN titrates away chaperones, and that promotes the heterologous aggregation, and there is evidence for that. There's a lot of cytoskeleton proteins have been identified that affect aggregation in yeast. These proteins prevent the initial aggregation that is a ring that shows up in the induction of Psi. These proteins, deletions in these proteins, prevent the ring from actually giving rise to Psi plus cells. These same proteins affect aggregation of poly-Q, but what we have here is that if you block this, you get extra toxicity, suggesting that these little aggregates may be indeed the toxic species. In any event, also SLA2 and SLA1 are involved and in the maturation and appearance of Psi aggregates. We can use yeast to try to look at the cellular functions that are targeted by the toxic species. Again, the existence of the modifiers identified in yeast should be very useful. To identify specific self-seeding conformations, particularly of TDP43 that's associated with so many different types of diseases, by isolating seeds from different patients with different disease. And finally, in therapeutic approaches, isolating protective intragenic mutations in the proteins that prevent simultaneously expressed wild-type protein from aggregating, looking for heterologous proteins that rescue by overexpression, and by setting up a screening platform for drugs. So in the end, I want to acknowledge some of the many people in my lab that have worked on this project over the years, my higher organism collaborators, and my mentors and colleagues.
Thank you. We have time for 2 questions. One over here. 3 questions, okay.
So a compelling idea from studies of aneuploidy suggests that altering the stoichiometries of proteins that normally form complexes can reveal, prion-like behaviors. So I'm curious if you have, is sub-35 prion formation, or really formation of any prion, changed if you increase the amounts of interacting proteins, things that normally form complexes with those proteins?
Yes, so, I'm sorry. I was too fast in turning. Can you hear me?
Yeah.
Okay. Yes, we showed, for example, that if you overexpress sub-45 that interacts with sub-35, that you reduce the prion formation. So,
Okay.
yes.
So the on-target complex
Yeah.
doesn't form.
Randy?
This is a comment, not a question, but just to amplify the last point that you made about using yeast as a screening vehicle. You certainly know that Sue Lindquist used yeast to screen for agents that would mitigate toxicity of expression of the mammalian alpha-synuclein. The inhibitor that they found is a stearate inhibitor of an enzyme, stearate desaturase, and that inhibitor now works in animal models, and it's in phase 1 clinical trials in humans.
Wonderful.
So this has been a great vision that she had.
So what is the reason for the different types of prions, you think?
They have different conformations. They have different folding conformations, and that's heritable. What do you mean, what's the reason for that?
No, that's what I meant.
Okay.
How do they arise? Yeah, from different conformations.
At random, when you overexpress, you get different types. And what we want to look for is different types, conformations of TDP43 that may cause different diseases associated with those types. But once they were called strains in PRP, so we called them, strains didn't work in yeast, so we called them variants. But now you guys use variants when you mean alleles. So they're self-seeding conformations.
Last question there.
You mentioned that you see both small and bigger foci with TDP43, but what's exactly the difference? Is it a structural difference, a compositional difference, or are they just
This is what we
size-wise?
want to test soon. So there's a recent paper showing small and large aggregates of TDP43, and one of them has, the small ones have very rapid FRAP, suggesting that they're liquid gel, and the large ones don't, suggesting that they're the solid aggregates. And we think that the ones we're seeing are probably like that. But we want to use that FRAP experiment, plus Randy Halfman has a FRET experiment where you can look at FRET changing in relative to protein composition that can also distinguish solid aggregates, oligos, and liquid gel. And we want to correlate that with the modifiers that we have that affect toxicity and see which species is the toxic species as best we can.
I'm afraid we have to move on, but we're going to hear more about prions from Reed Wickner.